In pathology, you can encounter many different problems. For several of those problems, scoring systems are available. And many of those scoring systems can be approached with image analysis. But which image analysis, which computer vision task is appropriate for which pathology problem? This is what you're going to learn in this video. Hi, I'm Alexandra Żuraw and I'm here to help you do better digital pathology. So if this is what you're after, be sure to subscribe, click the bell below, and be notified every time I release a new video. This video is part of a webinar I gave for the C.L. Davis Foundation. This was part of a day seminar and I was honored to be one of the speakers. I divided this webinar into smaller pieces so that they're easily digestible, but if you want to watch the full thing, you'll find the link below, and there's also going to be a playlist that you can binge watch if you want to watch everything. One of the biggest challenges in tissue image analysis is choosing the correct computer vision task for your pathology problem. I call it computer vision to pathology vision translation. And this is what this video is about, exactly what computer vision tasks should be used for what type of pathology problems. So let's dive into it so that you don't have to question yourself ever again.
I call it computer vision to pathology vision translation in quotation marks. So in computer vision, when we see everything and we don't have to think about, okay, do we only classify it, or maybe we should segment it, or maybe we should segment all the objects together. So computer vision is the part of computer science that tries to mimic human vision. So one of the challenges is, okay, how do you split the task of human vision? Because we just see, but they did. And let's start with something that resembles the human vision the most. This would be, so we have 3 giraffes here. We know where they are. We know that they are giraffes, and we can see that, like, where each one is beginning and ending, more or less. And this is instant segmentation. So we're able to recognize where they are. We can count them. We can see their borders. And then we can also, so in computer vision, there's also, you don't have to distinguish them from each other. You can just say, these are the giraffes, and here they are. Here are the borders of those giraffes as a group. And this is semantic segmentation. And then we can maybe say, okay, here's giraffe 1, here's giraffe 2, and here's giraffe 3, but we don't know the borders of those giraffes, but we know that this giraffe fits in this box. And this is object detection. So we can detect each giraffe, but we don't delineate it. And then we can just localize and classify that these animals, here are animals, they're here all together, and they're giraffes. And this is classification and localization. So for each of these tasks, we may have different neural network architectures. And I don't know which neural network architecture is for which, but I don't need to know this. What I need to know is how my pathology problems correspond to these problems in computer vision, because later either the software that I'm using has this already incorporated, or the computer scientist I'm working with is going to understand this language. He will know the computer vision if I tell him, okay, this pathology problem is this computer vision problem. So what do we want to do with our samples, our images, from the image analysis point of view? So when we look at our images, basically, we have either structures of interest or we have tasks of interest. So task of interest is, what do we want to do with those structures of interest? And structures of interest, this can be regions or objects. And for us, regions is going to be tissue. In veterinary medicine, it is very common to have multiple tissues on the same slides. And we can also have tissue compartments. So for example, tumor epithelial region versus stromal region. And objects, obviously we start with cells, but this can also be subcellular elements like vacuoles, like mitochondria, anything that is an object. Obviously, the most obvious for us are cells. Then we can also have non-cell objects like those beta amyloid plaques. They're delineated objects, but they're not cells. Or we can have objects made out of cells, but we don't really care about the cells, but we care about the whole object. For example, the glomerulus here, the renal glomerulus. So the tasks that we're interested in are our analysis endpoints. So when we want to use a tool for image analysis, we have to think, okay, what do we want to do with this image? What do we want out of this analysis? And this is gonna determine which of the computer vision tasks are we tackling. So yeah, what do we want to know about the objects, our objects or regions? One thing we might want to know is object identification and counting. And I love this picture. It's from Modern Pathology in 2015. This was the official recommendation how to count Ki-67 positive cells for breast cancer diagnosis. You were supposed to take a picture of an image, print it out, take a pen and mark in red the Ki-67 positive and mark in blue the non-positive, obviously only epithelial tumor cells. I love it. It's already ancient, 2015, because the field has developed so much and we have many algorithms that are approved for that. Not deep learning, just classical machine learning, but I love it. But basically here, we want to count Ki-67 positive cells. So what do we need? We need the correct numbers of those Ki-67 positive cells. Ki-67 is a proliferative marker, and this is an IHC stained image. What do we not need? We don't need the borders of those cells. We don't care. We just want to count them. So that was the first task. The second will be region localization and quantification. So in this example, we want to distinguish tumor epithelium versus tumor stroma. And this might be, for example, in pancreatic cancer, it is a prognostic marker, how much stroma there is between the epithelial cells. And this is a common endpoint because sometimes you want to quantify something just in one region, common image analysis endpoint. So what do we need here? We need correct boundaries because we don't want half of the tumor be in our stroma compartment. That's not what we want. Especially this is very much used in immuno-oncology, because we want to know where the immune cells are. Are they in the stroma? Are they infiltrating the epithelium? What's happening there with different populations of immune cells? And we also want correct area. We just mentioned that the amount of stroma, so the area of stroma in the side, might be a prognostic marker for some tumors. So we want to know the correct area. What do we not? We do not need to delineate those separate tumor islands. This is not of interest for this particular problem. But what do we do if we want to have objects identified, localized, and quantified and counted? We might want to have that. For example, when we do neuropath, we often quantify the numbers of neurons and calculate the G ratio. You can see on this image with the circles yellow inside. It's the ratio of the inner radius of the neuron to the outer radius of the neuron. It basically gives us information about the thickness of the myelin sheath. So for this, what do we need? We need everything. We need the exact boundaries of all those. So we need to count them. We want neuronal counts, because if we have less neurons, it's bad, obviously. Then we want to have the delineation of the outside of this myelin sheath, of the inside of the myelin sheath, so that we can then later compute the radiuses and calculate our G ratio. So for this particular problem, we need everything. So how do we translate computer vision tasks to the pathology tasks? This is, I call it computer vision to pathology vision translation. Object detection would be our Ki-67 counting problem. Then giraffes, just as giraffes, and that's fine. We don't have to distinguish one from the other. That's semantic segmentation, and that would be our tumor stroma separation. And then each giraffe exactly where it is, exactly with the delineation. This is instance segmentation, and this would be applied to the neurons. You may say, why don't we always do instance segmentation? If it has everything, we should always do it. Well, unlike our brain and eyes, computers are not that efficient. It basically costs a lot more computational power to deliver this kind of data than just those bounding boxes. So if we don't need to go and use the heavy machinery, why should we have to pay for this? And it's always a cost to benefit ratio.
Now that you've watched this video, you will always know which computer vision task should be used for which pathology problem, right? And if not, go back and just take a screenshot of where I explained this. But thanks so much for staying till the end, and be sure to watch some more videos.